Hello, everyone. Welcome to Global IMMUNOTALKs. My name is Asya Rose, and I'm the host today. As you all know, Global IMMUNOTALKs is a program that started during COVID by Carla Rotlin and Elina Zoniger. And since then, it opened the stage to explore the best researchers in immunology and who come and share the stage to give their talks. And today we have Wendy Barclay. Wendy is going to join us. Hello and welcome. And so
hello. Thank you for having me.
Great to have you. And we also would like to introduce you to our audience. Few words about Wendy. Wendy, she's the chair of infectious disease at Imperial College London. She graduated from Cambridge University, and for her postgraduate work, she actually infected volunteers with common cold viruses. But then she went on for her postdoc, where she did in the University of Reading and Mount Sinai Center in New York, where she was doing more molecular virology and using genetic manipulation on different influenza RNA viruses. And then in 95, she returned to the UK and started her work and continued working on these influenza viruses. Until in 2007, she moved to Imperial College, College in London, and she's now the head of the Department of Infectious Disease. And as we will hear from her, but her work really focuses on the mechanism by which viruses cross from animal sources into humans and cause new pandemics. Extremely visionary choice of research, which obviously during COVID, she was instrumental in also in the governmental way, in advising the government in the UK how to cope with this thing that came on us. And she was awarded by the CBE for services in virology and the New Year's Honors List of 2022. So we are very lucky to have you today. And in the tradition of this Global IMMUNOTALKs, we have a question for you. Can you describe a turning point in your career that defined your current research focus?
Yes, thank you. Thank you for the question. I'm going to talk about it today, this turning point in my career as part of the talk, but I just want to get across the excitement really of it. We had been trying to work out for so many years why these avian influenza viruses don't just cause pandemics all the time, and what was underlying this barrier for animal viruses to really infect humans productively. And we came up with a strategy. I have to say that I tried to persuade several different people in my lab to take this project on. It was almost the project of doom, and nobody wanted it because it was difficult. But in the end, one very brave postdoctoral fellow took it on. And I will never forget, at around about midnight, receiving an email back in 2015 from Jason, who I'll refer to in my talk today, saying, "We found it, we found it, we found it, we found it, we found it." And it just went on and on, this email. He was so excited. And I knew at that point that he was confident and I had confidence in him. And everything after that just took off for me. And we were able to pursue that and enjoy really picking apart what was going on at the molecular basis there. So, yeah, I think everybody hopes in their career they get one moment like that. And I'm so grateful that it happened to me and really brought the science alive.
It's amazing. I can totally understand. So we're looking forward to hear your talk and about this moment. So please, the stage is yours.
Thanks very much. I will hopefully share my screen. I'm going to choose this one and share. Can you just confirm?
Yeah.
It's good.
Looks great. Yes, please.
Okay, great. Okay, so I will begin the story. What I want to talk to you today is about how influenza viruses cross the species barrier to cause pandemics. So thank you very much for having me. And I guess I start off with a a slide or a statement which is perhaps more gloomy than you might want today, which is to say that just because we recently, around 5 years ago, experienced this coronavirus pandemic that we call COVID, does not make it any less likely that there will be another pandemic. And I guess as an influenza virologist, I would argue that influenza viruses remain amongst those which should be at the top of our list in terms of the risks. We know that viruses that infect animals can cross into humans and cause pandemics. And these schematics here of the coronavirus and up here of an influenza virus particle, these cartoons just illustrate that these are two of the classes of viruses that can do that. And by studying them in detail and understanding this crossing of species, hopefully we can learn more about how to tackle them, how to predict these pandemics, and even think about other viruses with that potential. So I'm talking today about respiratory virus pandemics. Of course, there could be pandemics that spread in other ways, but we know that when viruses spread through the air, they're particularly problematic to control. A feature of all pandemic viruses is that they will look different to our immune systems. And I've written that here as novel antigenicity. What I mean by that is that the human population will not have any existing immune memory, either B cell or T cell, of this virus. And so everybody is vulnerable, and the virus can spread amongst humans in an uncontrolled way. That's why you get such an explosive outbreak, because everyone exposed is potentially the next host. If it's a respiratory virus pandemic, then the requirement for the virus would be to replicate efficiently in cells of the human airway, because this is how the virus will move from one person to the next. And the third ingredient on the list is that it can indeed do that. It can survive in airborne particles and transmit between us in those respiratory droplets that we exhale. And finally, I've put a fourth one on here, which I don't necessarily think is essential, but I observe that if transmission occurs before symptoms and before people know that they are infected and contagious to others, then the initial phase of the pandemic really becomes very difficult to control. And I refer here, just as an example, the difference probably between SARS coronavirus 1, which caused horrible outbreaks and horrible disease, and then SARS-CoV-2 may well be that SARS-CoV-2, the cause of COVID, transmitted so much more efficiently, and in particular from people who didn't even yet know that they were infected. And I would argue that influenza viruses that have caused pandemics in the past can also do this. So what my lab likes to focus on is how do pandemics arise, which virus of all of the different animal viruses out there and all the strains of influenza in particular, which one of those is most likely to cause the next pandemic, and in the hope that by understanding all of these, we could change our practices and maybe prevent some pandemics. And I use influenza as the best example, really, for a pandemic virus. There are many, many influenza viruses which exist in numerous wild animal reservoirs, both in birds and also in bats. Amongst those animal reservoirs, there are at least 19 different antigenic subtypes to which humans have no prior exposure. And I would also say that pandemics can perhaps come to humans more readily when they transit through a source, a different animal, an intermediate source, perhaps domesticated animals with which we have larger interface. And we know for influenza virus that the poultry that we farm, there's chickens and ducks, and also pigs, and even other domesticated animals can all become infected with influenza and potentially pass their virus on to humans. So there is opportunity for the virus to cause a pandemic. There is potential because there are many antigenic subtypes. And not only that, but we know that influenza viruses have caused pandemics in the past. And here we have just a map, if you like, of the 20th century and the early 21st century, illustrating the 4 pandemics that came to humans from animal sources during those 110 years or so. At the moment, the world is perhaps teetering on the edge of a new influenza pandemic. There is a particular subtype, an antigenic subtype of influenza known as the H5N1, which emerged and caused much concern at the latter part of the 20th century, but has continued to infect both humans and animals around the world. And we are in the middle of what is known as a panzootic, where there are abundant and widely globally distributed H5N1 viruses, which are infecting birds and mammals and sometimes humans. And this has once again raised alert to the potential of this virus to cross from those animal sources into people and cause explosive outbreaks. This particular subtype H5N1 is split, as are all flus, into many complicated phylogenetically defined clades. This one is known as 2344B. And very sadly, these highly pathogenic viruses are often lethal in birds and have caused die-offs of very important wild bird populations around the world. They have crossed into sea mammals, such as seals and sea lions, into domesticated animals that probably were fed contaminated poultry, and even into farmed mammals here, farmed mink being farmed for fur. And the most recent event, which is causing concern, is the spillover of this virus from birds into dairy cattle, a host which has not been previously described as an influenza virus, an influenza A virus host. But in early 2024 in the United States, signs of milk production declining, contaminated milk, alerted dairy farm workers and public health officials, and eventually a test was done for this H5N1 virus, which was certainly in the wild birds and poultry in the area, and indeed these the mammary glands of these cows were infected with this virus. And of course, there is a huge interface between humans and these animals when we milk them. And several, more than 70, in fact, human cases of this clade 2344B H5N1 influenza virus have occurred across the United States, most of them linked to exposure to cows or to chickens infected with the now cow virus. Interestingly, most of these are associated with mild disease, in contrast to the zoonotic infections that were noted in the earlier part of the 21st century, when the H5N1 viruses first began to spread, but nonetheless causing concern. Here you can see a dairy farm worker who reported with conjunctivitis. In people perhaps with some level of compromising of their immune system, there have been more severe illnesses and one death. So the question that we're all asking around the world is, how worried should we be about this H5N1 influenza virus that is in dairy cattle, and indeed about the H5N1 influenza viruses which are out there in other birds and mammals around the world. And here today I'm representing a group of researchers from the United Kingdom who are funded in a consortium to think about how do these viruses cross from birds into mammals, and how close are they to causing pandemics? So what I need to emphasize is that there is not an H5N1 pandemic at the moment. And that's because, as I will go on to explain, there are many molecular barriers which halt the virus on this journey that it could potentially take from being an avian virus spread through the water between ducks and geese to a zoonotic virus that perhaps infects a person exposed to very high levels of virus, could cause severe disease, could cause mild disease, but these are often dead ends and don't go any further. Sometimes an influenza virus will then acquire sufficient mutations, which I will talk about in detail today, which will transform it from what is essentially a bird virus into a virus which can replicate in the cells of the human airway and spread between humans. And then, of course, those viruses that cause pandemics don't go away, as we now see with COVID, the SARS-CoV-2, and with all of the influenza viruses that we've documented in outbreaks in the 20th and 21st century. Such viruses transform into seasonal viruses, being driven forward in evolution by our own immune responses. So antigenic drift, meaning that we have to update vaccines to keep pace with virus evolution, and further human adaptation, optimizing transmission between us. So not all avian influenza viruses can do this. Hardly any of them actually do. And the ones that do will be those who those which have opportunity and that can support and undergo a set of mutations which cross from these avian influenzas into those with the pandemic pandemic capability. And the incompatibility of all of those first avian influenza viruses that are not adapted, the incompatibility of those avian influenza viruses with humans occurs at two levels, which I'm going to split out into two halves of my talk today. The first is this issue of replicating inside the cell. All viruses are obligate intracellular parasites. They have to bind their host cells, they have to gain access to machinery which is inside the healthy host cell and co-opt it for themselves, and they have to avoid a bunch of innate restriction factors which otherwise would suppress their replication. And avian influenza viruses have co-evolved with their bird hosts for many years and are very good at doing this in avian cells, but the factors which they need to gain access to successfully replicate inside mammalian cells may well be different. And this would mean that there's an incompatibility which slows the virus down and lays it vulnerable to other restriction factors. And even if an influenza virus or any virus can overcome that barrier, then that would be enough to result in a severe case of zoonotic infection of a human with an animal virus. But a pandemic would only ensue if that virus were then capable of passing from one person to the next and to the next and to the next with an R value greater than 1, in other words, that it continues to spread through the community. And avian influenza viruses, as I've intimated, are essentially viruses which have evolved to spread through the gut of birds and then through the water on which these migrating waterfowl land as they migrate and share their water. And the lifestyle change, if you like, to become a human respiratory virus is really rather different. In that second instance, the virus needs to be capable of surviving in airborne droplets and in human respiratory secretions and passing from one person to the other through the air. And unsurprisingly, therefore, there are sets of changes which this avian influenza virus would need to undergo in order to have an efficient transmissibility. So first of all, inside the cell, as I've already said, the virus needs to get inside the cell. Influenza viruses bind to sugars known as sialic acids, which are displayed all over the surface of the cells of our noses and throats. They enter the cell through endocytosis and they release their genome into the infected cell nucleus, where it grabs hold of many different proteins and factors and carries out its replication and expresses all of the proteins of the virus. And eventually all of these new synthesized genomes and viral proteins come together and make new viral particles. All the while, the virus is also needing to evade our own innate immune system and other restriction factors which may be constitutively expressed that might otherwise inhibit its replication. And the outcome of infection is the balance between the access and compatibility with all of the pro-viral host factors required and the ability of the virus to evade the antiviral factors. And this is the case for every virus, and for influenza virus, what we like to do is to flesh out the detail of that and understand what are all those different host factors that impact the virus inside the cell, how are they different between birds and mammals, including humans, and what are the mutations in the viral genes that help optimize this virus-host interaction. So the first part of my story is going to be about the polymerase enzyme of the virus. Like many viruses, influenza carries its own polymerase into the cell with it. The polymerase is made of 3 proteins known as PB1, PB2, and PA that come together as a heterotrimer and bind to the RNA. The RNA genome of the virus is a segmented genome. There are 8 different pieces of RNA in every virus particle, which constitute all of the genetics encoding the viral genome. And the RNA is always complexed with a nuclear protein. And together, the RNA, the nuclear protein, and the polymerase are known as the viral ribonuclear protein complex. And this is what the virus carries into the cell as its replicating unit. Once inside the nucleus of the cell, this polymerase can first of all transcribe the viral RNA into messenger RNAs, each one encoding a different protein of the virus, which are then translated inside the cell by the ribosomes and made into all the different proteins. And the viral RNA is also replicated via a full-length complementary intermediate into new genomes, which will form part of the new virus particles, which complex together with the proteins that have been synthesized. Now, inside a human cell, if an avian influenza virus is capable of delivering its genome to the nucleus, one of these steps doesn't happen. And in fact, it's this step, the replication of the genomes, is deficient. And what that means is that most avian influenza viruses cannot productively replicate inside human cells. Now, you would be saying, well, how can this be? Because humans have been infected by avian influenza in the past, and indeed you've told us that we've got human influenza viruses, which once upon a time derived from a bird virus, but now, of course, spread quite happily amongst us in our seasonal influenza outbreaks. And the reason for that is that although this is a barrier for the virus, it's not a very strong barrier. The influenza virus, as I've said already, has an RNA genome. RNA replication is inherently error-prone and unstable, and what that means is that RNA viruses evolve very quickly, and indeed mutations which give an advantage to the virus in a particular situation, like a new host, are rapidly selected. There is a single point mutation in one of the subunits of the polymerase, the PB2 subunit, which can switch on the ability of the avian virus polymerase to now replicate inside the human cell. A simple switch of charge from glutamic acid to lysine at this residue number 627. And this mutation is often seen in cases of humans who have been exposed to avian influenza viruses, perhaps in poultry markets, and they may have severe disease, and within several days, the virus that they're replicating inside their lungs is all switched to this mutation. And indeed, in the earliest of the pandemics that have been properly documented, the famous 1918 or Spanish influenza pandemic, we can see that that virus had an origin originally in birds, but already at the time of causing the pandemic has this adaptive mutation in its polymerase, enabling it to replicate to high titers in humans. And indeed, that virus, that mutation is maintained through all the seasonal flu viruses of the 20th century. And in fact, because the influenza virus genome is segmented, that switch not only is readily achieved by a single point mutation within a host, but can also be readily adapted, if you like, by this process known as reassortment, whereby if a virus already has that PB2 adaptation and is happily replicating in human cells, but it finds itself replicating in the same cell as an avian virus, then during the assembly process of the new virus particles, this gene shuffling or reassortment can happen. And now a new virus can emerge, which is already adapted to replication in human cells, but has acquired a new antigenicity by acquiring the spike proteins, or the H and the N, the proteins that adorn the virus particle from the avian virus. And we know that this is how the pandemics of 1957 and 1968 in the 20th century arose, by this reassortment event. So it's a barrier, it's a very interesting barrier. It's clearly a very robust barrier for the virus, but the virus can overcome it either by point mutation or by this process of reassortment. But for many years, we wanted to understand more about the nature of the barrier. And we had two hypotheses. We wanted to know whether or not these mutations that are seen in the viral polymerase of human viruses compared to those that originate in birds were there to enable the virus to evade a human restriction factor, or were they there to enable the use of a human ortholog of an essential pro-viral host factor? And this is my exciting result. This is Jason Long, who was a postdoc in my lab and was persuaded to take on this crazy screening project where we obtained a bunch of hamster hybrid cells with parts of the chicken genome in them. And there were many different clones of these hamster chicken hybrids. And we infected them, or we reconstituted the avian influenza polymerase inside all of these different clones. There were 50 or so different clones, and each one contained a different piece of chicken chromosome, in addition to the hamster chromosomes. And we reasoned that if there was one gene in the chicken genome which could support the replication of an avian influenza virus, then some of these clones would be expressing that chicken gene in addition to all the hamster genes they were expressing, and they would become permissive for the avian influenza. And indeed, when Jason performed this mini replicon assay, in which we can measure the activity of the viral polymerase by recording the luciferase produced from an artificial viral-like RNA, what he could see is that the introduction of just one single chicken gene, the chicken ANP32A gene, was sufficient to switch on the activity of that avian virus polymerase. The introduction of the human ortholog of that gene did not. So we, by a series of further experiments, showed that this species difference in this ANP32A gene between birds and mammals underlies this influenza A virus polymerase host restriction. ANP32 is a protein which you can find in human cells, in swine cells, in bird cells. Of course, it's a host protein. It's not there for the benefit of influenza virus. We haven't evolved to have a protein like that so we can become infected by influenza. The ANP32 proteins are a family of proteins which are important for the healthy host, and they bind histones and control transcription. And they have this interesting structure where they have a series of leucine-rich repeats, and then this unstructured region here, which we've just made up, which is a low-complexity acidic region. As I've said, there's a family of ANP32 proteins. You can find such proteins present in the nucleus of all mammals and birds that are acting as hosts to influenza. There are 3 functional family members, ANP32A, B, and E, and they have important roles in the healthy host. But what was important for us was to understand, indeed, was there a difference between the ANP32A proteins that are expressed in bird cells and those which are expressed in mammals? And indeed, we could find it. We could find it at the DNA level, in that the ANP32A gene has undergone a level of gene duplication, which creates an additional exon. And that means that a splice variant of ANP32A can encode a protein which is 33 amino acids longer than the mammalian equivalent. And that 33 amino acids turns out to be an insertion in the structure of the protein between the leucine-rich repeat and the low-complexity acidic region. And you can see that all of these flighted birds have this gene duplication, so it must have occurred at a point when after the common ancestor, if you like, branched off to the flighted birds. But what was absolutely fascinating, and why Jason and I got so excited, was that the ostrich, which of course is a bird, did not have this extra exon and therefore only encoded a short version of AMP32A. Now, why on earth am I talking to you today about ostriches? I'm so excited about those. Well, it turns out that ostriches can become infected by avian influenzas. And what had been noted, without really knowing why, was that when that happens, mammalian-like mutations in the polymerase subunit PB2 were selected for as the virus circulated through ostriches in a farm, for example. And in other words, when an influenza virus finds itself inside the cell of an ostrich, it behaves as if it was inside the cell of a mammal. And that told us that the host factor, which was determining these mutations and determining this host species restriction at the level of the polymerase, was indeed this AMP32 gene. So to sum up that story, what we can say is that avian influenza viruses encode a polymerase with a glutamic acid in PB2, and they can't infect us because they can't co-opt proviral factors, AMP32 proteins, which are present in mammalian cells. They have co-evolved, indeed, a polymerase which can co-opt this longer avian AMP32A protein. But this species barrier can be overcome by this single charge switch, a point mutation of a glutamic acid to a lysine, which now enables the human adapted virus to co-opt the AMP32 proteins which are present inside human cells. And we now know in molecular detail how this is all working. This orange blob in the middle here represents the three-dimensional structure solved by cryo-electron microscopy of the AMP32 protein, and it is bridging two polymerase units. This first one here in blue is associated with the RNA template, and this is the one that's going to carry out replication. And the second one here in green is known as the encapsidase. And as new RNAs are synthesized by this polymerase, this encapsidase is going to be placed onto them to form the viral ribonucleoprotein complex. And then, in fact, the nucleoprotein will be recruited also by this AMP32 low complexity acidic region and placed on there as well. Now, this LCAR in the cryo-EM structure is not solved. We don't know where it is, but we suspect that it dangles down here between the two polymerases. And marked in bright red here is residue number 627, both on the replicase and the encapsidase. And you can see how that low complexity acidic region may well be placed between those two. If this is an avian influenza polymerase, these two are glutamic acid amino acids, which are themselves acidic. And the 33 amino acids that is inserted inside between the LRR and the LCAR would then displace that charge clash. On the other hand, if this is a mammalian or a human adapted polymerase, these glutamic acids have now been substituted for lysines. And that in its own right also undoes the charge clash, because now you have a positively charged residue against the negatively charged acidic region of the host factor. So this beautiful replication complex explains the host restriction and how this single point mutation can overcome it. So, as I've said, it really does only take a single point mutation to overcome the host restriction, and in many of the hundreds of people that have been infected by avian influenza viruses, particularly those studied since 1997, the viruses that they incubate already have acquired the polymerase adaptation. And yet, as I emphasize again, those viruses, H5N1, H7N9, and other avian influenza viruses that have infected humans have not sparked, as of yet, a pandemic. And what this tells us is that this adaptation of polymerase to be able to replicate in human cells is necessary, but not sufficient for a pandemic. So what indeed is that other host barrier which is saving us currently from influenza pandemics? The polymerase adaptation is a prelude that enables further adaptation in other viral genes that would then enable the virus to be transmitted through the air. And for the rest of the seminar, we're going to be thinking about transmission of viruses in airborne droplets, as represented by this rather wonderful cartoon. And of course, as I've said already, the lifestyle switch for an avian influenza virus, which is spread through the fecal-oral route and through large patches of water, to become a virus which survives in respiratory droplets and spreads from one person to the next, is quite different. And the ecology change, if you like, is going to be determined by what's on the outside of the virus, the structural components of the virus. And the two key proteins we'll talk about are the spike proteins of influenza, the hemagglutinin, or the H, and the neuraminidase, or the N. These are the proteins which interact with the external environment. The first fact that has been described for many years is that the receptors, which the hemagglutinin, as the receptor binding part of the virus, sees, are quite different between birds and humans. They are all sugars, they are sialic acids, but birds express abundant forms of alpha-2,3 linked sialic acid, which have a rather long and thin shape. And humans express abundant alpha-2,6 linked sialic acid, which is a much more globular molecule. And you can see here on this beautiful staining from Yoshi Kawaoka's lab, which has been parts of human airway stained with lectins which seek out either of these sialic acid linkages. You can see how predominant the alpha-2,6 linked receptor is on the apical surface of the human airway. In other words, when an avian influenza virus enters the human airway, there are hardly any cells to which it can attach. And what this means is that it can't transmit. And we showed this, as have many other people. We used our reverse genetics technique here, where we took a virus, a human H3 virus, which binds to the alpha-2,6 sialic acid and transmits between humans and also between ferrets, which are a very good model of human transmission because they also have abundant alpha-2,6 sialic acid in their nose and throat. And we showed that this human virus could transmit from one infected animal to a naive sentinel with 100% effectiveness. If we back mutated the hemagglutinin of that virus at just 2 positions to switch its sialic acid binding from the alpha-2,6 to the alpha-2,3 linked sialic acid, the virus no longer transmitted. We were able to infect animals with this virus if we used a high dose of virus. And by performing what's known as an ID50, a dose titration, we were able to establish how much virus was actually needed to initiate infection in these animals. And you can see that almost 40-fold more virus was required to initiate infection if it was binding the wrong receptor. So, to put this another way, if you imagine a ferret or a human expelling virus particles into the air, their concentration will diminish as the virus floats through the air in airborne droplets, and then the amount of virus reaching the next animal or the next person could be quite low. If that amount of virus is lower than the amount required to initiate infection, then no transmission will occur. And that's what happens if the virus is binding to the wrong form of sialic acid receptor. All of the human infections with H5s and H7 viruses so far maintain this preference for the avian-like receptor, and this switch has not occurred. In contrast, the pandemics of the 20th century, H1, H2, and H3, all show these hallmark mutations in the hemagglutinin, which switch on the ability to bind to the abundant sialic acid receptors in human nose and throat. Once hemagglutinin binds to the surface of the cell through this sialic acid, it enters endosomes, and in the acidic environment of the endosome, it undergoes this famous mutation, this famous fusogenic transformation, where the shape of the molecule completely transforms into a fusion machine, enabling the virus to fuse its membrane with that of the endosome and release its genome. This conformational change is triggered by the acidic environment inside the endosome. But it turns out that the triggering pH is different for human viruses and for many avian viruses. And these avian viruses, like the H5N1 viruses, which have been causing so many problems, have a much less stable hemagglutinin. In other words, they more readily undergo this irreversible and fusogenic switch. If this switch happens on the outside of a cell, it is irreversible and the virus is no longer infectious. So influenza viruses can be inactivated in acidic environments. You can see, interestingly, that human influenza viruses have this pH of fusion at 5.5 or lower, meaning that they undergo this switch much less readily, but on the other hand, are much more stable in acidic environments. The environment of the human respiratory tract is mildly acidic. This very nice review from Charlie Russell pulled together some measurements that have been made over many years by different groups. But you can see that healthy adults and children have a nasal cavity which is at a relatively low pH and very close to the pH at which an avian influenza virus would undergo that irreversible inactivating change. And we were able once again, to use our reverse genetics technique to create a pair of viruses which only differed by a single amino acid that made the pH of fusion of the hemagglutinin lower. And once again, the human influenza virus is infected into the ferret. It transmits with 100% efficiency between one ferret and the next. The avian influenza virus, which is destabilized just in the hemagglutinin, does not transmit. And if we measure the infectious virus in airborne droplets using this custom-made piece of machinery here, where the infected animals breathe, their droplets bearing their virus pass over the surface of open plates and can be collected and quantified. What you can see is that the stable virus survives in the airborne droplets all the way to the meter distance at the end of the plate in infectious form, whereas the destabilized virus with the avian virus hemagglutinin, which falls apart, if you like, at this low pH, does not survive beyond the first plate. And even those viruses we find on the first plate turn out to actually be mutant or revertant viruses which have restabilized. So this emphasizes once again the importance of this hemagglutinin switch. Two different genetic changes in hemagglutinin required to switch receptor preference and maintain pH stability. And a final barrier, even if the virus reaches a person in sufficient amounts and still infectious, is that the nose and throat of the human airway, the apical target cells, are lined with respiratory mucus. And for many respiratory viruses, including influenza, this respiratory mucus is a barrier. But actually, influenza virus has a neuraminidase, a sialidase enzyme on its surface, which can cleave through the mucus and allow it to desialate the decoy receptors, which would otherwise block the virus from finding its receptors on the real target cells below. And we and others showed that the neuraminidase enzyme of many avian influenza viruses is also structurally different than that of those which are human transmissible. Very often, the avian neuraminases have an enzymatic head which is protruding from the surface virus particle a shorter distance, because there have been amino acid truncations here selected for in the avian hosts. And we were able to set up this assay, which is a little bit like a neutralization assay, where we harvest airway mucus from the surface of airway epithelial cells, we incubate it with infectious virus, and then we ask how much infectivity is recovered by the activity of the neuraminidase enzyme, which is present on the virus particles. And here you can see a human transmissible virus, which is very effectively recovering its infectivity. And here you can see an avian virus with only a short stalk neuraminidase, and it can't recover its infectivity. And if we're simply using our reverse genetics, again, restore the length of the stalk, some infectivity is recovered. So if I add up everything I've told you so far, the good news is that many avian influenza viruses that have some degree of pandemic potential do not cross into humans or intermediate hosts and then go on to cause pandemics, because there are many host range barriers which the virus would have to overcome by accumulating mutations in several different virus genes. The first one is this polymerase restriction, which is actually rather readily overcome. But after that, and seemingly much more tricky for the virus, is the switch of receptor binding, the balancing of the neuraminidase, and the maintenance of enough stability of virus particles that it remains infectious in airborne secretions. So not very many viruses are out there with the opportunity to accumulate all of those mutations and transform into airborne transmissible pandemics, which is why we don't have more frequent influenza pandemics than we might otherwise have. So having told you all of this, we are now in a situation where we can go back to the cows, which are currently causing concern, and ask ourselves, where on that journey, on that staircase, is the virus that is in the dairy cows in the United States? How worried should we be? So the bad news, first of all, is that we can already see, as we might expect, that the polymerase genes of this dairy cow virus have got some mutations in them, not perhaps the ones that we might have expected, but they are indeed mutations which can confer the ability on this virus to co-opt the human and the bovine ANP32 proteins. So this mutation that we've discovered here at position 631 in the PEB2 gene lies very close to the interface between the polymerase here in yellow and the host factor here in orange, and almost certainly allows the virus to utilize the ANP32 genes of its new host, the cow, and unfortunately of the humans as well, which is why it is able to cross from cows into humans and replicate inside those individuals who've been exposed. Some more bad news is that unlike the earlier H5N1 viruses of the early part of the 21st century, the new dairy cow virus has a neuraminidase with a longer stalk, and this can quite effectively cleave through human mucus. So here's our mucus neutralization assay again. You can see the human transmissible virus is fully capable of cleaving human mucus. The old H5N1 viruses are not and do not transmit. Now, if we look at the neuraminidase gene of these new viruses which are circulating in the world today, they are able to cleave human mucus. And if you give that neuraminidase gene to otherwise transmissible viruses, it's capable of supporting airborne transmission. So why don't we have a pandemic from the dairy cow virus? Because, thankfully, the dairy cow virus hemagglutinin remains acid unstable and does not bind to human receptors. So here our collaborators, Jeon and Munir in Pirbright Institute in Surrey in the United Kingdom do this biolayer interferometry assay. You can see in red, the cattle virus does not bind to the human form of sialic acid receptor, and it binds extremely well to the avian form of receptor, the opposite pattern than a human transmissible virus. However, we around the world and many people are monitoring the changes which might be selected for in these dairy cow viruses as they continue to circulate between cows. And this phylogenetic tree here shows the hemagglutinin gene of the H5N1 hemagglutinin that's in the virus in cows. And there are a number of interesting mutations cropping up and being maintained in cows. And if you map those onto the three-dimensional structure of the H5 hemagglutinin, you can see here that some of them map extremely closely to the sialic acid receptor binding interface. The big worry is, are these mutations going to switch the receptor preference from alpha-2,3 linked sialic acids to alpha-2,6, which would be one step along the pathway that we would be very uncomfortable about in terms of the pandemic threat from these viruses. Of course, that evolutionary drive will rather depend on what kinds of sialic acids are expressed in the mammary gland of a cow. And in order to address that, Jack Hassard and Stuart Haslam at Imperial College have been performing glycomics of the mammary gland of cows. So what this is a mass spec analysis of the sialic acid receptors which are present in those tissues. And if you sum up what this tells you, what it tells you is that there is an abundant amount of sialic acid there. But interestingly, the form of sialic acid present in cows is a form known as NuGUC. NuGUC is different than NuAC, which is the form of sialic acid which predominates, for example, in humans. NuAC is converted into NuGUC by an enzyme known as CMAR, and it turns out that CMAR is not functional in humans. What we can see when we perform the biolayer interferometry assays with synthetic analogues of the NuGUC receptor is that those mutations that we described that are arising in cows and being maintained are switching up the affinity of the virus to use this receptor that is present in cows, known as NuGUC. And the good news is that, therefore, the virus is adapting to better replicate in cow mammary tissue, but none of these mutations have enabled binding to the human receptor, the 6-SLN or 2,6-sialic acid, nor have they made it more airborne transmissible in terms of pH stability. In fact, cows, therefore, are unlikely to select for the kinds of changes that will result in the next pandemic, as far as we can see at the moment. That doesn't mean to say we should dismiss the worry. We should keep monitoring very, very carefully to see what's happening as this virus is now almost 2 years into its circulation in these new hosts. And meanwhile, we should be very careful about this panzootic, which is ongoing, of the clade 2344b influenza viruses of H5N1. They have already found their way, for example, into mink farms and other fur farms in Europe and in the United States. And perhaps one might envisage that it's in these kinds of animals, which are very similar to our favourite experimental model for human-to-human transmission, the ferret, that the most worrying adaptive changes may occur. So I will finish there with this long list of acknowledgements to the wonderful people in my group, past and present, who've contributed to the data I've shared with you today, our many, many collaborators who form wonderful structural biology, who look at things from the veterinary and One Health point of view, and of course our funders. Thank you very much for having me on your IMMUNOTALK seminar series, and I do hope you've learned something new today. Thank you.
Thank you very much, Wendy, for this really interesting and very timely, a bit scary talk. So, and as you all know, you can ask questions, and I'm sure there are going to be many of those on the YouTube as comments. So please use this opportunity and engage in this discussion. I also want to mention that next week is a talk on global immunotox. Is going to be Ilev, and we hope to see you there as well. So thank you very much, Wendy, again, and thank you everyone for joining us today.